Hello friends, welcome back to the lectures on diabetes. So today we shall be discussing about the pathogenesis of type 1 diabetes and type 2 diabetes. Myself, Dr. Santosh Abraham, working as a specialist registrar in endocrinology and diabetes at Scarborough General Hospital. So briefly we will discuss about type 1 diabetes and type 2 diabetes, the pathogenesis. Under type 1 diabetes we will discuss about genetic susceptibility, about HLA and non-HLA genes influence, environmental factors and mechanism of beta cell destruction. And the type 2 diabetes will talk about genetic factors, environmental factors, metabolic defects, insulin resistance and its mechanism, obesity and insulin resistance, and beta cell dysfunction. And finally we'll discuss about the pathogenesis of complications, both acute and chronic, and then we will go on to the histopathology of pancreas in diabetes, hyaline arteriosclerosis, diabetic microangiopathy, and diabetic nephropathy. So let's start with the pathogens of type 1 diabetes. As you all know, type 1 diabetes is an autoimmune disease. Here there is islet destruction, and it is caused by immune effector cells which react against the endogenous beta cell antigen. So it's an autoimmune disease directed against the beta cell antigens. The disease can develop at any age, including adulthood, and the previously used appellation juvenile diabetes is now considered inaccurate. Also, we all know that it was called insulin dependent diabetes before, and this has been excluded from the current classification of diabetes because all forms of diabetes may be treated with insulin. Genetic susceptibility. There is a higher concordance rate for the disease in monozygotic versus dizygotic twins, and this has been convincingly been established in type 1 diabetes. Moreover, genome-wide association studies known as GWAS have identified multiple genetic susceptibility loci for type 1 as well as for type 2. What about HLA Association? More than 30 susceptible loci for type 1 are now known. Of these, the most important locus is the HLA gene cluster, which is located on chromosome 621. According to some estimates, this contributes as much as 50% of the genetic susceptibility to type 1. Approximately 90 to 95% of Caucasians with type 1 have either an HLA-DR3 or HLA-DR4 haplotype. Also, 40 to 50% of the patients with type 1 diabetes are combined DR3 DR4 heterozygotes. Individuals who have either DR3 or DR4 concurrently with a DQ8 haplotype, which corresponds to a DQA10301, DQB10302 allele, demonstrate one of the highest inherited risk for the type 1 diabetes in sibling studies. But it is not still known whether these HLA disease associations reflect the ability of the specific HLA molecules to present self-islet antigens, or if they are related to the role of HLA molecules in T cell selection and tolerance. The influence of non-HLA genes. Several non-HLA genes also confer susceptibility. The first one was the gene identified for insulin itself, were the VNTRs in the promoter region associated with the disease susceptibility. These polymorphisms may influence the level of expression of insulin in the thymus, thus affecting the negative selection of insulin-reactive T cells. Other associations are seen with polymorphisms in CTLA4 and PTPN22. It has to be emphasized that there is a good relationship of type 1 diabetes to the altered T cell selection and regulation, and individuals with rare germline defects in genes that code for the immune regulation, such as the AIRE, which also cause the autoimmune polyendocrineopathy syndrome APS type 1, show prevalence of this disease, that is show prevalence of type 1 in these individuals who have the AIRE mutations. What about the environmental factors? It has been proposed that antecedent viral infections could function as triggers, but there is no definite proof for that. One suggested hypothesis is that viruses might share epitopes with islet antigens, and the immune response to the virus itself in cross-reactivity and destruction of islet tissues, something known as molecular mimicry. The molecular mimicry that might explain the influence of viral infections. But infections are also known to be protective against type 1 diabetes. How is the beta cell destroyed in type 1 diabetes? The fundamental immune abnormality is a failure of self-tolerance in T cells specific for islet antigens. And this failure of tolerance may be due to the combination of defective clonal selection of self-reactive T cells in thymus, and also due to defects in the function of regulatory T cells or resistance of effector T cells. In short, autoreactive T cells not only survive but are poised to respond to self-antigens. Initial activation of these cells is thought to occur in the peripancreatic lymph nodes. It could be in response to antigens that are released from the damaged islets. This activated T cells then move to pancreas, where they cause beta cell injury. Multiple T cell populations have been implicated, including Th1 cells, CD8 plus CTLs. So the Th1 cells secrete cytokines, including interferon gamma and tumor necrosis factor, that injure B cells, while the CD8 population kills beta cells directly. The islet autoantigens, which are the targets of immune attack, include insulin, beta cell enzymatic glutamic acid decarboxylase, also known as GAD, and ICA512. The presence of islet cell antibodies is used as a predictive marker for this disease, but it is not clear if the autoantibodies cause injury or are merely produced as a consequence of islet injury. Now let's come to the pathogenesis of type 2 diabetes mellitus. So type 2 diabetes is a complex disease, and it involves an interplay of genetic and environmental factors with a pro-inflammatory state. But there is no evidence of an autoimmune basis for type 2. The genetic factors are genetic susceptibility contributes to pathogenesis. So it's evidenced by the disease concurrence rate of greater than 90% in monozygotic twins. Also, first degree relatives have a 5 to 10 fold higher risk of developing type 2 diabetes. GOAs have already identified at least 30 loci that confer a minimal to modest increase in lifetime risk for type 2 diabetes. The most important environmental risk factor is obesity, particularly central or visceral obesity. So greater than 80% of the individuals with type 2 diabetes are obese, and incidence of diabetes has risen in proportion to obesity. So this contributes to the cardinal metabolic abnormalities of diabetes and insulin resistance. What are the metabolic defects in diabetes? The two cardinal defects are: one is insulin resistance and other is beta cell dysfunction. So beta cell dysfunction means inadequate insulin secretion in the face of insulin resistance and hyperglycemia. What is insulin resistance? So insulin resistance is the failure of target tissues to respond normally to insulin in relation to a given glycemic load. The liver, skeletal muscle, and adipose tissue are the major tissues where insulin resistance is manifested, and it results in the following. It results in failure to inhibit endogenous glucose production in the liver, so it further results in high fasting blood glucose levels. It also leads to failure of glucose uptake and glycogen synthesis in skeletal muscle following a meal. So it contributes to high PPBS or postprandial blood glucose, and also failure to inhibit lipoprotein lipase in adipose tissue leads to excess FFAs, which in turn amplify the state of insulin resistance. In short, failure to inhibit endogenous glucose production, failure of glucose uptake and glycogen synthesis, and failure to inhibit lipoprotein lipase results in increased fasting blood sugars, postprandial blood sugars, and excess free fatty acids respectively. So what are the mechanisms behind this insulin resistance? There is reduced tyrosine phosphorylation of the insulin receptor and IRS proteins in the peripheral tissues. This compromises insulin signaling and reduces the level of GLUT4 on the cell surface. An interesting observation is that exercise can improve insulin sensitivity through increased translocation of GLUT4 to the surface of the muscle cells. What is the relation between obesity and insulin resistance? The risk for diabetes increases as the body mass index increases. Not only the absolute amount but also the distribution of the body fat that has an effect on insulin sensitivity. We all know that central obesity is more likely to be linked with insulin resistance. Free fatty acids. There is an inverse correlation between fasting plasma FFAs and insulin sensitivity. And central adipose tissue is more lipolytic than peripheral sites. This might explain the particularly deleterious consequences of this pattern of fat distribution. What happens when there are excess FFAs? They overwhelm the intracellular fatty acid oxidation pathways. This results in the accumulation of cytoplasmic intermediates like diacyl glycerol DAG. These are toxic intermediates and these can attenuate signaling through the insulin receptor pathway. In liver, usually insulin inhibits gluconeogenesis by blocking the activity of PPCK, the first enzymatic step in this process. When there is attenuated insulin signaling, it allows PPCK to ramp up gluconeogenesis. And excess FFAs also compete with glucose for substrate oxidation, and hence it leads to feedback inhibition of glycolytic enzymes. Thereby it further makes the existing glucose imbalance worse. What are the role of adipokines in insulin resistance? So adipose tissue is a functional endocrine organ and it releases hormones in response to change in the metabolic status. A variety of proteins are secreted by the adipose tissue, and these are collectively called adipokines or adipose cytokines. Some of these promote hyperglycemia, whereas others decrease blood glucose, such as leptin and adiponectin. Leptin and adiponectin, they decrease the blood glucose by increasing insulin sensitivity in the peripheral tissues. In obesity, adiponectin levels are reduced, and thus they contribute to the insulin resistance. Inflammation. Diabetes is now known to have an inflammatory milieu which is mediated by pro-inflammatory cytokines that are secreted in response to excess nutrients such as FFAs and glucose. This results in both insulin resistance and beta cell dysfunction. Excess FFAs within macrophages and beta cells activate the inflammasome. This is a multi-protein cytoplasmic complex which causes secretion of the cytokines interleukin 1 beta. The IL-1 beta in turn mediates the secretion of additional pro-inflammatory cytokines from macrophages, islet cells and other cells. So inflammation, how this causes beta cell dysfunction or the insulin resistance. So it is mediated by the pro-inflammatory cytokines, and excess FFAs within the macrophages and beta cells activate inflammasome, which leads to IL-1 beta secretion, and IL-1 beta secretion causes additional pro-inflammatory cytokines to be secreted from macrophages, islet cells and other cells, thereby exacerbating the insulin resistance. IL-1 and other cytokines are also attributed. They are released into circulation and they act on major sites of insulin to promote insulin resistance. So they are also attributed to insulin resistance. Thus, you can see that excess FFAs can impede insulin signaling directly within peripheral tissues as well as indirectly through the pro-inflammatory cytokines. Now about beta cell dysfunction. The insulin resistance leads to impaired glucose tolerance, while the beta cell dysfunction is virtually a requirement for the development of overt diabetes. Without the beta cell dysfunction, the diabetes will not be formed, or it will not lead to the development of diabetes without beta cell dysfunction. So this dysfunction actually increases early in the disease. The beta cell function actually increases early in the disease. That is, the beta cell will start working more so as to compensate the insulin resistance and thus maintain euglycemia. But eventually the beta cells exhaust their capacity to adapt to the long-term demands of peripheral insulin resistance, and the hyperinsulinic state gives way to a state of relative insulin deficiency. So this is how the beta cell dysfunction makes diabetes manifest. Now the mechanisms of beta cell dysfunction. Several mechanisms have been implicated, but the following four are the most important. One is excess free fatty acids, which result in lipotoxicity. The impact of chronic hyperglycemia called glucotoxicity, an abnormal incretin effect in which there is reduced secretion of GIP and GLP-1, and finally amyloid deposition. So the mechanisms are due to lipotoxicity, glucotoxicity, decreased incretins and increased amyloid deposition. The monogenic forms of diabetes are discussed under genetic factors in type 2 diabetes mellitus and also in a subsequent presentation on MODI. So you are advised to watch that. So let's come to the pathogenesis of complications. Now talking about the acute complications, the most important one is DKA in type 1. So many of the factors which precipitate DKA, like illnesses and starvation, are associated with the release of catecholamine epinephrine. This blocks any residual insulin action and it stimulates the secretion of glucagon. The insulin deficiency coupled with the glucagon excess decreases peripheral utilization of glucose and it increases the gluconeogenesis. This leads to an exacerbation of hyperglycemia, and the hyperglycemia causes an osmotic diuresis and dehydration, which is very characteristic of the ketoacidotic state. Second major effect of insulin deficiency is activation of the ketogenic machinery in DKA. It stimulates lipoprotein lipase when insulin is deficient, with resultant breakdown of adipose stores, and it results in an increase in FFAs. When these FFAs reach the liver, they are esterified to the fatty acyl coenzyme A, and oxidation of fatty acyl coenzyme A molecules within the hepatic mitochondria produces ketone bodies, which I've already told you before in another presentation. In contrast to type 1 diabetes, the frequency of ketoacidosis is significantly lower in type 2 because of the higher portal vein insulin levels. This prevents unrestricted hepatic fatty oxidation. fatty acid oxidation, and keeps the formation of ketone bodies in check. But type 2 diabetes may develop a condition known as HHS due to severe dehydration resulting from sustained osmotic diuresis. So HHS has got a different mechanism. It is due to sustained osmotic diuresis, whereas ketoacidosis forms due to the deficiency of ketones and due to the defects in oxidation in the liver. So the most common acute complication in either type of diabetes is hypoglycemia. It usually results by missing a meal or excessive physical exertion or excess insulin administration. Now let's come to the pathology of chronic complications of diabetes. We all know that the morbidity associated with either type is due to damage induced in large and medium sized muscular arteries, diabetic macrovascular disease, and also in small vessels called the diabetic microvascular disease. This is all due to chronic hyperglycemia. The macrovascular disease will cause accelerated atherosclerosis. Pathogenesis of chronic complications. Glucotoxicity is responsible for the long-term complications. There are 4 distinct mechanisms through which this persistent hyperglycemia causes these deleterious effects. In each of these proposed mechanisms, there is increased glucose flux through various intracellular metabolic pathways, and it generates harmful precursors that contribute to end organ damage. In short, there is increased glucose flux and it generates harmful precursors which results in end organ damage. So let's examine what are these. The first mechanism is formation of advanced glycation end products, also known as AGE. They formed as a result of non-enzymatic reactions between intracellular glucose derived precursors with the amino acids of both intracellular and extracellular proteins. And AGE leads to release of cytokines and growth factors, generation of ROS or reactive oxygen species, and increased procoagulant activity in endothelial cells. So the ROS and the procoagulant activity is increased in endothelial cells, and finally it results in enhanced proliferation of vascular smooth muscles and synthesis of extracellular matrix. The second mechanism is through the activation of protein kinase C. So activation of protein kinase C and second messenger diacylglycerol DAG is an important signal transduction pathway. Intracellular hyperglycemia stimulates the de novo synthesis of DAG from glycolytic intermediates. Thus it causes excessive PKC activation, and the downstream effects of PKC activation are numerous. It could include the production of VEGF, TGF beta, and PAI1, that is a plasminogen activator inhibitor 1, and these are all produced excessively by the vascular endothelium. The third mechanism is oxidative stress and disturbances in polyol pathways. So nerves, lenses, kidneys, blood vessels generally don't require insulin for glucose transport, but persistent increased glucose concentrations in the extracellular environment leads to an increase in intracellular glucose in these tissues, and this excess glucose is metabolized by the enzyme aldose reductase to sorbitol, which is a polyol, and eventually to fructose in a reaction that uses the NADPH. NADPH, is also required to regenerate reduced glutathione, GSH. So reduced glutathione is an important antioxidant mechanism in the cell. Any reduction in the GSH increases the cellular susceptibility to reactive oxygen species. Thus, progressive depletion of intracellular NADPH by aldol reductase will compromise the GSH regeneration. Thus, the cellular susceptibility to oxidative stress increases. Sorbitol accumulation can also cause cataract formation in the lens. The last mechanism is hexosamine pathways and generation of fructose 6-phosphate. So hyperglycemia induces hexosamine pathway, and then it increases the intracellular levels of F6P, that is fructose 6-phosphate. It is a substrate for glycolization of proteins. It means that it will lead to increased excess proteoglycans. And what are the effects of these proteoglycans? These excess glycosylation changes are accompanied by the abnormal expression of TGF-beta or PI1, which will further exacerbate the end organ damage. Now let's come to the histopathology in pancreas in diabetes. So in pancreas we can see that there is a reduction in the number and size of islets. Mostly it's seen in type 1 diabetes. There are leukocytic infiltrates in the islets composed of T lymphocytes, so it is called insulitis. And in type 2 diabetes there may be a subtle reduction in the islet cell mass. Another characteristic feature of type 2 diabetes is amyloid deposition in the islets, and there is an increase in number and size of the islets in type 2 diabetes. So in reduction in number and size of islets in T1DM, while there is an increase in number and size of islets in type 2 diabetes with amyloid deposition, while there is a leukocytic infiltration called insulitis in type 1. So you can see insulitis here in type 1, and here you can see the amyloidosis in type 2. So both have a different picture on histology. Another important factor is hyaline arteriosclerosis, which is usually associated with hypertension. So this is another important histological finding. It is more prevalent and more severe in diabetes, but it is not specific for diabetes. It presents as a form of amorphous hyaline thickening of the wall of arterioles. This causes narrowing of the lumen. In diabetics, it is related not only to the duration of the disease but also to the level of blood pressure. So both of these will cause an increased hyaline arteriosclerosis. That means if you have the blood sugars unchecked and the blood pressures high. So this is how the hyaline arteriosclerosis looks like. And coming to the pathology of diabetic microangiopathy. So this is one of the most consistent morphological features, and it is due to the diffuse thickening of the basement membranes. It is most evident in skin capillaries, the skeletal muscle capillaries, the capillaries of retina, renal glomeruli, and renal medulla. It could be also seen in non-vascular structures such as renal tubules, the Bowman capsule, peripheral nerves, and placenta. Now coming to the pathology of histology of diabetic nephropathy, there are 3 lesions commonly encountered. 1 glomerular lesions, 2 is renal vascular lesions, and it is principally arteriosclerosis there. And the third one is pyelonephritis, including necrotizing papillitis. Let's discuss about the most important glomerular lesions, and they are the most important here. So the most important glomerular lesions are: 1 is capillary basement membrane thickening, 2 is diffuse mesangial sclerosis, and 3 is nodular glomerulosclerosis. Diffuse mesangial sclerosis shows a diffuse increase in mesangial matrix, and it is associated with overall thickening of the globular basement membrane, and the matrix depositions are PAS positive. The progressive expansion of mesangium has been found to correlate with the proteinuria. Nodular glomerulosclerosis, which is another important pathological finding in long-standing diabetes. It is also known as Kimmel-Wilson disease or intercapillary glomerulosclerosis. The lesions are ovoid or spherical and they are often laminated, and there are nodules or matrix situated in the periphery of the glomerulus, and these nodules are PAS positive. They lie within the mesangial core of the glomerular lobules, and all the glomeruli may not be affected. They can be surrounded by patent peripheral capillary loops or loops that are markedly dilated, and they may produce microaneurysms as the untethered capillaries distend outward due to the increased pressure. As the disease advances, the individual nodules enlarge and may eventually compress and engulf the capillaries, and this obliterates the glomerular tuft. These nodular lesions are frequently accompanied by accumulations of hyaline material in capillary loops called fibrin caps, or adherent to the Bowman capsules, also called capsular drops. So the hyaline material may be in the capillary loops called fibrin caps, or it could adhere to the Bowman capsules called capsular drops. Both the afferent and efferent glomerular hyla arteriole will show hyalinosis. As a consequence of glomerular and arterial lesions, the kidney suffers from ischemia, and it develops tubular atrophy and interstitial fibrosis, and usually undergoes an overall contraction in size. Thanks for watching this presentation.